We will start with Golgi apparatus. The second part under the topic of endomembrane system. In the previous session we have completed endoplasmic reticulum. Smooth endoplasmic reticulum, when it gets detached, it helps in formation of Golgi apparatus. Now this Golgi apparatus, it was first given by Camillo Golgi. In the year 1898. Now he observed these structure like a densely stained reticular structures at the peripheral side of the nucleus, and he called them as Golgi bodies, and they are named after the scientist name. Now these members, they are formed by the detachment of smooth endoplasmic reticulum. So these are synthesized by smooth endoplasmic reticulum. They are mostly observed in the eukaryotic cells. In eukaryotic cells. We are saying it as mostly. Why? Because there are certain exceptions are present. So what are those exceptions? Except mature sieve tubes. In case of plant cell, that is mature sieve tube cells that does not consist Golgi apparatus. And number 2, mature RPC cells, or else mature mammalian RPC cells that does not consist of Golgi apparatus. Along with that, sperm cells. The sperm cells of bryophytes and pteridophytes. They also does not consist of Golgi apparatus. Now apart from these things, there are some more special cases are present. Now this Golgi apparatus, they consist of similar structure when compared to endoplasmic reticulum. They also consist of cisternae, vesicles and tubules. But the cisternae of Golgi apparatus in case of plants, they are also sac-like structures, but they don't have interconnected system. So here cisternae is not interconnected. In case of plants, and such Golgi apparatus is called as dictyosomes. It means if question is asked, presence of dictyosomes, it's a characteristic feature of, that is, plants. Now this is one special property. We'll go for the second. In case of fungi, it consists of very poorly developed Golgi apparatus, where the cisternae is very less developed, and that is called as unicisternal Golgi bodies. It's a characteristic feature of fungi. If you look at the Golgi apparatus, it consists of 4 parts: cisternae, vesicles, tubules. Along with that, one additional structure is present, that is called as Golgian vacuole. Now here is a cisternae, and the cisternae is having many interconnected sac-like structures which ranges between 5 to 8 in number, and towards the periphery it gets pinched off and helps in formation of vesicles. It consists of 2 phases. The lower side, it is called as cis phase, which is near to the endoplasmic reticulum, or else it is also called as convex phase, or it is also called as forming phase. And towards the opposite side, that is called as trans phase or concave phase, or else it is also called as maturation phase. Now whatever the materials that gets received from the cis phase, they get transported and gets packed and released through the trans phase. So here is the cisternae, vesicle, and here is a Golgian vacuole. Now let's go on to the functions of Golgi apparatus. Now this Golgi apparatus, it helps in packaging, processing and transporting of materials from cis phase to trans phase we said, which are getting collected from the endoplasmic reticulum. Now that becomes the first function. Number one, it helps in processing, packaging, and transport. They help in processing, packaging, and transport of materials from cis phase to trans phase. Number two, it acts like an important site for the synthesis of glycoproteins and glycolipids. Glycoproteins and glycolipids. And that is by a process called as glycosylation and glycosidation. If you talk about glycoproteins, it is by glycosylation. Glycosylation of proteins that leads to formation of glycoproteins, and glycolipids are formed by glycosidation, and that is from lipids. Now these Golgi apparatus, they are rich in root cap cell, due to which it secretes certain mucilaginous substances and helps in lubricating the root tip, due to which the root penetration will be easier and there won't be any damage that is caused to the root apex. Number three, it is rich in root cap and it helps in lubrication of root tip. In case of head of sperm cell, it consists of modified Golgi apparatus, and that is called as acrosome. So here modified Golgi body in head of the sperm cell, that is called acrosome. Now whatever the materials that are getting transported from endoplasmic reticulum, in the diagram you can see the materials or else the proteins which are getting secreted from the endoplasmic reticulum, they are received from the cis phase and gets modified and transported and get converted into enzymes, and these enzymes they get filled into the Golgi vacuole and becomes into primary lysosomes. It means what happens here? Primary lysosomes are synthesized from the trans phase of Golgi bodies. So what is another function here? It helps in synthesis of primary lysosomes. It also helps in formation of cell plate during the time of plant cytokinesis, and it also helps in plasma membrane formation when during the time of cytokinesis. So most of the times you will be getting question from the point of functions. They are very, very important. In the endomembrane system, many a times we have got a question. So especially that is from the point of functions. In the endoplasmic reticulum, in the Golgi apparatus, you might expect a question based on the functions.